Welcome to the SmartSoleHub Podcast. I'm Jae Kim. If a peripheral nerve is physically trapped and generating pain, why does injecting a simple 5% dextrose solution right next to it actually stop the pain? For a long time, the main explanation for perineural injection therapy, or PIT, was purely mechanical. We thought the fluid was just physically hydrodissecting or pushing the compressed tissue away from the nerve. But here is the clinical paradox. I use a superficial subcutaneous technique in the clinic. I'm not always achieving a massive mechanical release deep at the exact entrapment site, yet my patients are still getting profound relief. That means the dextrose isn't just a physical wedge, it is a biologically active molecule. To understand what dextrose is doing chemically, we need to look at what happens when a nerve gets trapped in the first place. When a nerve is compressed, its local blood supply is restricted. This creates local ischemia. The nerve is literally starving for both oxygen and glucose. Nerves are incredibly metabolically active structures. When they are starved of energy, the cells signal distress by releasing pro-inflammatory cytokines, specifically a molecule called TNF-alpha. In the short term, this inflammatory response is meant to recruit repair, but in the long term, it keeps the nerve highly sensitized, inflamed, and locked in a chronic pain state. Which brings us to a 2022 paper by Wu and colleagues published in the journal Life. They wanted to see exactly how glucose interacts with these inflamed nerve cells at a molecular level. They took a human cell line that mimics peripheral nerve cells and cultured them in a petri dish. Then they exposed them to TNF-alpha to simulate a severe ischemic nerve injury. After the cells were thoroughly inflamed for 16 hours, the researchers bathed them in various concentrations of glucose to see what would happen. They found three specific things. First, metabolic recovery. As expected, the TNF-alpha exposure caused the cell's mitochondrial function to drop significantly. The cells were struggling to produce energy, but when treated with a 25 millimolar glucose solution, the metabolic function recovered. The starved cells were given a concentrated energy substrate, and they began to function normally again. Second, the inflammatory output dropped. The initial stress had caused the cells to release massive amounts of interleukin-6 or IL-6, which is a major inflammatory marker. The 25 millimolar glucose treatment significantly suppressed this release. But the third finding is the true breakthrough, and this is where I want to use a specific metaphor to explain what is happening. The researchers looked at a cellular protein complex called NF-kappa B. I want you to think of NF-kappa B as the master switch for inflammation inside the cell. When this switch gets flipped on, it commands the cell's DNA to start mass-producing pain enzymes and inflammatory proteins. The researchers proved that 25 millimolar glucose significantly suppressed the activation of that NF-kappa B master switch. The dextrose isn't just feeding a starved cell, it is actively reaching into the cell's internal pathways and turning off the master switch for neuroinflammation. Reading this paper was a massive aha moment for me. It provides a clean biological rationale for why superficial injections can calm an inflamed nerve without needing a heavy mechanical release. But, as always, we have to be honest about the evidence hierarchy. This is a level 5 in vitro study. We are talking about isolated cells in a highly controlled laboratory environment, not a complex living human foot that takes 10,000 steps a day. It gives us a beautiful molecular framework, but it doesn't independently prove clinical outcomes. And that leads me to a massive clinical puzzle that kept nagging at me while reading this. It comes down to the dosage. The researchers saw these incredible anti-inflammatory benefits at exactly 25 millimolar of glucose. But in the clinic, we inject a 5% dextrose solution. If you do the math, 5% dextrose is roughly 277 millimolar. That is 11 times higher than the dose used in this study. The researchers actually addressed this discrepancy. They justified their 25 millimolar test dose by assuming that when we inject a syringe of 5% dextrose into a patient's tissue, the fluid dilutes roughly tenfold as it diffuses and spreads out over the next few hours. That is a very reasonable assumption, but it is just an assumption. Nobody has actually measured the real-time glucose concentration right next to a nerve after a subcutaneous PIT injection. So, what happens if the concentration at the nerve ends up being 50 millimolar or 100? We know that traditional prolotherapy uses huge concentrations like 15 or 25% dextrose specifically to trigger severe inflammation and force tissue repair in ligaments and tendons. So the response curve clearly isn't a straight line. A moderate amount of glucose suppresses the NF-kappa B master switch, but a massive amount of glucose triggers inflammation. I genuinely don't know where the exact clinical sweet spot sits. If we increased our PIT injections from 5% to 10% dextrose, would we deliver more healing energy to the nerve, or would we accidentally push past the threshold and cause a flare-up? The study doesn't answer that, but what it does prove, without a doubt, is that concentration matters immensely, and it confirms that perineural injection therapy is a profoundly biochemical treatment, not just a mechanical spacemaker. To sum it all up, when a nerve is starving and inflamed, a precise dose of glucose doesn't just feed it. It actively turns off the master switch for neuroinflammation, allowing the nerve to finally rest. You can find the full breakdown of this paper, my clinical notes, and diagrams of the NF-kappa B pathway over at jaekim.com.au/blog. Thanks for listening. Next time, in part 4, we are going to tackle the biggest clinical paradox of them all. Why does chronic high blood sugar in diabetes destroy nerves, while dextrose injections heal them? I'll see you then.